Urine formation takes place in 3 steps. First one is the glomerular filtrate, which is a process by which the blood that passes through the glomerular capillaries is filtrated through the filtrate membrane, what we studied above. And the other one is the tubular reabsorption. Reabsorption of what? They absorb only the selective substances. The absorption takes place in 2 methods. One is the active and the passive. In active, with the help of ATP, materials are like minerals are absorbed from the blood. In passive, without any use of ATP or energy, the absorption of urea, water and chloride is taken place. Different parts of our nephron will absorb different amount of materials, like glucose is absorbed 100% in the proximal convoluted tubule. Amino acid is also for 100%. Maximum amount of sodium is also absorbed in the proximal convoluted tubule. Calcium, phosphate, water 70%. Whereas in the proximal straight tube, that is nothing but the end of the proximal convoluted tube where the phosphate absorption is seen. In the thick ascending limb and the Henle's loop, sodium, potassium, calcium and magnesium absorption is seen. Distal convoluted tubule is also thick, and the thick ascending limb is also thick, which are not permeable for even water. This reabsorption is activated with the help of ADH hormone. Here ADH hormone is acted on this so that the permeability of these tubules is increased, so we can absorb calcium, magnesium, water and all the things. In collecting duct, depending upon the concentration, the absorption is taken place. Normally, the regulation of the tubular reabsorption is done by 3 techniques. One is the glomerular balance, hormone factors and the nervous. Hormone factors like antidiuretic hormone, aldosterone, parathormone. Parathormone is used for the reabsorption of the calcium, magnesium and the hydrogen. Aldosterone is to reabsorb the sodium. Antidiuretic hormones are used for the absorption of the water. And then last one is the tubular secretion. Normally, kidneys play a very important role in the maintenance of the acid-base balance. How they maintain? By secreting the hydrogen ions and by reabsorption of the bicarbonates. Hydrogen ions are like acidic in nature. Bicarbonates are basic in nature. For example, in our body, there are about 4,380 MEQs hydrogen ions are formed. Bicarbonates are about 4,320. To maintain this balance, about 50 to 800 MEQ of hydrogen ions are secreted through the urine. Right? Hydrogen ions are secreted through the 2 mechanisms. First one is the sodium hydrogen antipode and the other is the ATP driven proton pump. ATP driven proton pump is nothing but with the help of ATP, hydrogen ions are excreted out through the tubules. Right? And in the sodium hydrogen pump, in the exchange of the sodium and hydrogen will be excreted and the sodium is reabsorbed. Role of kidney in preventing the metabolic acidosis. First one is the bicarbonate mechanism. In the bicarbonate mechanism, when the bicarbonate enter into the lumen or when it is filtrated, it combines with the hydrogen ion to form the carbonic acid. This carbonic acid is divided into the carbon dioxide and the water. These two will enter the tubular cells. Again, there it fuses to form carbonic acid. Again it divides to bicarbonate and the hydrogen. This hydrogen ion is again absorbed into the tubular lumen and the bicarbonate is absorbed into our body, that is in the interstitial fluid. And the other part is second one is the phosphate mechanism. In the phosphate mechanism, the hydrogen which is secreted into the renal tubule reacts with the sodium hydrogen phosphate to form the sodium dihydrogen phosphate, which is secreted through the urine out. Ammonium mechanism. Ammonia is also formed in the tubular cells. is secreted into the tubular lumen in the exchange for the sodium ion. In the same manner, this is also excreted through the urine. Hydrogen ions are excreted in the place of ammonia. Renal functional test. There are two types of test. First one is to measure the GFR, where the known amount of the inulin material which is not reabsorbed nor secreted is injected into our body, and then we have to see or examine the urine, how much volume of urine is excreted, plasma concentration and the concentration of the inulin in the urine with the help of formula UV by P, where U stands for the concentration of the substance in the urine, V stands for the volume of the urine flow, and P stands for the concentration of the substance in the plasma. Right? For the measurement of the renal plasma flow, we use para-aminohippuric acid with the same formula. This is also material which is not absorbed but excreted through the urine. So we can easily examine this.